Good afternoon. It's my pleasure to introduce today's wall speaker. By way of introduction, I'd like to point out that according to a recent survey, the number of macromolecular structures in the protein data bank is growing at a rate of 8,000 a year. One group of proteins, however, remains vastly underrepresented in this expanding encyclopedic knowledge of structures, and that is the class of membrane proteins. At the end of last year, 2010, there were a total of only 789 deposited membrane protein coordinates representing 279 unique membrane protein structures. Today, you're in for a visual treat, and we'll hear from one of the world's foremost authorities on membrane protein structures, Professor Johann Deisenhofer. Dr. Deisenhofer has been a professor at the University of Texas Southwestern Medical Center at Dallas since 1988. Early in life, he became interested and fascinated, actually, with the infinite expanse of outer space and imagined himself to be an astronomer one day. Eventually, his interest shifted to the microcosmos, as you will learn, manifested in the arrangements of atoms in protein molecules. Born and educated in Bavaria, Germany, he received his master's degree in physics from the Technical University of Munich, his doctoral research in protein crystallography at the Max Planck Institute for Biochemistry and the Technical University in Munich. He continued his structural analysis of biological macromolecules by performing X-ray crystallographic studies during his postdoctoral fellowship and later as a staff scientist, both at the Max Planck Institute. Dr. Deisenhofer's arrival in the U.S. coincided with the announcement that he would share the 1988 Nobel Prize in Chemistry with his colleagues Hartmut Mikkel and Robert Huber for their structural analysis of the photosynthetic reaction center. This membrane pigment protein complex is the key molecule in the process of photosynthesis and has been described as mediating the most important chemical reaction on the planet Earth. Since then, his laboratory at UT Southwestern has remained at the forefront of the field and determined many incredibly complicated, high-profile membrane protein structures. His contributions to our understanding of membrane proteins are legion and have been recognized by awards too numerous to list, but many of them are actually listed in the handout that you've gotten. Although overshadowed by his achievement in structural membrane proteins, Dr. Deisenhofer's contribution to crystallographic methodology is by no means less significant. He and his colleagues pioneered the methodology of constrained crystallographic refinement for biological macromolecules, which is now a standard part of macromolecular crystal structure determination, and whose impact has gone beyond the field of macromolecular crystallography. And I would add that he has mentored generations, a couple of generations now, of structural biologists, including two very much admired members of our own faculty, Susan Buchanan and Dee Shah, who are both senior investigators here. Today, Dr. Deisenhofer will share with us his view on the future direction of structural biology. His talk today is entitled Structural Biology, Achievements and Challenges. Please join me in welcoming Dr. Deisenhofer.
Thank you. Good afternoon, and thank you all for coming. I am always flattered by such wonderful and kind introductions that we just heard. Sometimes I feel a little bit doubtful whether this is really about me. That is, it is me who is announced in this way, but it seems so. I recently closed my lab, so I'm in a post-PI phase of my existence, and therefore am confined to talking about more historical and speculative aspects of my research interest. So I will give you a very small, very short introduction of the history of structural biology and comment about membrane proteins and try a brief outlook on the future. And in the end, I want to concentrate on a topic that is right now close to my heart, and that is trying to completely understand proteins. And I venture the opinion that we are far from doing that. So let me start at the beginning. Here is a list of some historical events. They are probably all known to you. Rosalind Franklin, Jim Watson, Francis Crick, and of course the first crystal structure of a protein, or the structures, by Max Perutz and John Kendrew. And later on, new techniques came up. So X-ray diffraction was the first to give structural information. And I should mention, before these, there were, of course, other people like Northrop and Sumner who first crystallized proteins. And I especially like a small paper by Dorothy Crowfoot, as her name was at the time, and J.D. Bernal on how to get a diffraction pattern of a protein crystal, which was not possible without keeping the crystals moist. And that was a big practical step in making people really convinced that protein crystals are real crystals, because when they were treated as if they were salt, then they dried out before any diffraction could happen. And but this is a sort of half-page little story in Nature in 1934. Anyway, so this came before all these. And then the inclusion of electron microscopy into studies of biological objects by Aaron Klug, and then also by Richard Henderson, Nigel Unwin, who actually gave the first glimpse of a membrane protein structure by showing the 7 angstrom resolution structure of bacterial rhodopsin obtained from 2D, two-dimensional crystals. Then, of course, came Kurt Wittrich, who almost single-handedly introduced the power of NMR into studying macromolecular structure. And so, since then, major technical advances, in my opinion, were the ones, or among the major technical advances, were the ones listed there. And first of all, that people learned to manipulate DNA and to introduce DNA constructs into bacteria and have them express proteins. And I cannot overstress the importance of this, because otherwise, and I was already in the business by that time, we were sort of confined to studying proteins that naturally abundantly occur in an organism, and sometimes as a result of pathological circumstances, like the myeloma proteins, for example, which gave rise to the first structures of antibodies. And this development freed the whole field and made possible the study of many, many proteins. Then, of course, behind everything is the development of computers. Without this step, there would not be structural biology as we know it today. limited to X-ray crystallography, of course, the arrival of dedicated synchrotron X-ray sources was a big step, and nowadays we collect all our data on from such sources, or almost all. A seemingly tiny step, but in my mind also very important, was the ability to freeze crystals. For a long time it was considered impossible because the formation of microcrystals of water inside the protein crystal would destroy that, and this is also true. But if you do it in a certain way, quickly and with application of additives, then it works. And without that, a lot of the successes would not have been possible. And then also there's an enormous development in methods solving the phase problem, concatenating steps of crystallographic structure solution into software suites that run almost automatically. And so this is the result. These are the structures in the protein data bank as of this month, and you can see that right now we have these over 70,000 structures deposited. They're of course not all unique, but just to give a number, here in yellow are the annual additions, and they are in the order of 8,000. And of course, in the beginning, the growth was very slow. And I think the advent of recombinant of heterologous expressed proteins was about here, and then it was proven first that they are the same in structure as the ones that occur in the original or home organism, and from then on the field set off. And still most of the structures are done by X-ray crystallography. NMR has a very significant part of new structures, and electron microscopy is still a little bit behind, but I believe it is coming and gaining importance. It will gain importance in the future. A special case are, and these were already mentioned in the introduction, membrane proteins, and I like to show this slide because I think it was typical of a situation where everyone knew something that was wrong. And George Fehr is a very distinguished scientist who worked on photosynthesis, and he at some point in the late 1970s he felt tempted to try and crystallize a photosynthetic reaction center, and he applied for a grant, and the reaction was this. So luckily in Europe there were people who did not know this or didn't believe it, and one of them was Hartmut Michel, and this is a cartoon he made in the early 1980s, how one could imagine working on membrane proteins structurally, and of course they would have to be solubilized from the membrane by using detergents, and these are characterized here by this square with a single tail group, and as opposed to lipids with a double tail group. And just to this is just to indicate that the lipids are more likely to form bilayers, and the detergents are more likely to form micelles, spherical particles. And these would arrange around the hydrophobic surfaces of membrane proteins, which are shown here as full lines, whereas the hydrophilic surfaces would be are shown as dashed lines. And the task is to crystallize such complexes of detergent, and as the grant reviewer correctly mentioned. But Hartmut thought, so what? I mean, why not try that? And he imagined two types of crystals that could be formed. One, more similar to membranes, so the detergents would be replaced again by lipids in this example. And then these planar arrangements would come together in an ordered way, so to form a three-dimensional stack of membrane-like arrangements. The other one would be a detergent membrane protein complex, where the hydrophilic surfaces made the crystal contacts, and the detergents remained sort of in the open spaces between the protein molecules. And this is the type of crystal that he made in the early 1980s using photosynthetic reaction centers. And I had the privilege to join him in this endeavor, and together we produced this structure, which was already mentioned, the photosynthetic reaction center from something now called Blastochlorus viridis, at that time was Rhodopseudomonas viridis. And it consists of 4 protein subunits called L, M, H, and cytochrome, and a number of cofactors that are embedded in the proteins. Especially if you turn this 90 degrees, you see the peculiar shape of this central section, which is, as we know from many indications, inserted in the membrane. Not the last of the indications is that the surface of the protein in this area is totally hydrophobic, even more hydrophobic than the interior of soluble proteins. And so this structure had quite a positive impact on photosynthesis research, and especially the arrangement of the cofactors was kind of a topic for heated discussions between people in this field. They already used fast laser spectroscopy to try to follow the events in this reaction center after absorption of a photon. But because for lack of structural knowledge, many of the discussions ended in discussions about what might be the underlying structure of the absorbing molecules that were sort of causing the spectra. And for that reason, it had this unbelievable impact. And I can tell you a small story. The first time I traveled in America with having some essentially knowing this arrangement here, I gave a talk at the University of Oregon, and there were some strange people in the audience, and nobody knew them. And it turned out they had traveled from Seattle to Eugene, which is, I was told, a 5-hour drive. And I thought, okay, I must have done something really important, because who would drive 5 hours to just hear a seminar of somebody you don't know anyway? So that was the impact on photosynthesis research. And of course, over time, it turned out that not only this arrangement is important, but also the interaction of these cofactors with the protein. And here are histidines that bind to magnesium ions in the chlorophylls, and there are histidines binding to a single iron down here, and a glutamic acid. And all this is embedded in the L and M subunits. This is the L and this is the M subunit. And the effect of these proteins is that, or seems to be, that electrons starting out from a pair of chlorophylls in the middle move along a chain of cofactors on one side of this arrangement. This was the big surprise that the structure was actually providing. And so this is a copy of a slide I made a long time ago, which shows the sequence of events. Light hits a pair of chlorophylls. It takes about 2.8 picoseconds for an electron to move from an excited pair of chlorophylls to a neighboring molecule, which is chlorophyll-like, but called pheophytin. It doesn't have a magnesium in the center of its ring. Then it takes 200 picoseconds for the electron moving on to the quinone, a first quinone called QA, and then within about microseconds, the electron moves on to the second quinone. And this repeats again until the second quinone is fully reduced. It picks up protons and then moves out of the reaction center, is processed by the cytochrome BC1 complex, whose structure was solved here at the NIH in the JAS group by Lothar Esser. And, and this BC1 complex transfers the electrons back through the membrane, and they end up on the cytochrome and are fed back into where they came from. And the net effect is that proton pumping across the membrane, so 2 protons per electron, and that drives then the proton gradient across the membrane that drives all kinds of cellular process in this bacterium. Okay, the second effect of our work was that now nobody could say you cannot crystallize membrane proteins, and so grants were given and money was flowing into the field, and that had the effect that structures came forward. Here is a diagram made by Steve White a few years ago, and it shows the sort of the number of cumulative new structures in the protein data bank, and they follow an exponential law like this, n equals exponential at, where a is the sort of a time constant, and this time constant turned out to be 0.29 per year, and for soluble proteins, and 0.24 per year for membrane proteins. And if we consider that the 0 points of the two curves are 25 years apart, soluble proteins start in 1960 and membrane proteins in 1985, these structures were solved in a very, very different technological environment. At this point, for membrane proteins, it was already the beginning of the use of synchrotrons. Computers were infinitely better already. Many of the methods for crystallography had been developed or were in the beginning, whereas the people who did the first soluble proteins fought with very, very different circumstances. And yet, it's interesting to see how a simple exponential law can describe the rise of these structures. And nowadays, this is an up-to-date diagram for membrane proteins. This constant had to be slightly adjusted. It was 0.24 before, and now it's 0.23. It looks as if there was a time when a lot of, a bit more of membrane proteins were determined than expected according to this law. And maybe it's slowing down a little bit, but that probably is just statistical noise. And so we are going, but this number is of course very small in view of the fact that the human genome encodes for, or all the proteins in the human genomes, of all the proteins, 30% are membrane proteins. So there is quite a way to go. And I mean, one of my favorite membrane proteins nowadays is this, photosystem 1, published in 2001 by a group from Berlin, including Wolfram Sanger and Peter Fromme, who is now in US. And it is a real monster. It has 12 protein subunits, 127 cofactors, but the nice thing is that this is a trimer, so it looks a little bit bigger than it would need to be. Threefold axis is here, and the nice thing is that in the center of this there is a a structure that is similar to the structure of the bacterial photosynthetic reaction center. So as if this arrangement of 2 subunits with 5 helices each had been invented once and was considered an ideal solution to the problem. And also photosystem 2, which is the oxygen evolving photosystem in plants and cyanobacteria, has this arrangement of 5 helices each. And this is not very easy to see on this picture, but you just have to believe me. So a few remarks about what comes next in structural biology. I have to admit I don't know much about NMR, but I heard from some of my people who tried to or collaborated with NMR groups to solve protein structures that one of the main fields to catch up is automation. So they complained about the many things you have to do by sitting at your computer terminal and typing in things that should essentially be already known to the system and so on. So this is just refinement, and I cannot comment on any new methods that would be coming up in NMR. Electron microscopy, as I said, has a very promising future. One can now see, on the one hand, papers that report what they call sub-nanometer, that is, better than 10 angstrom resolution structures, and some of them already are in the order of 4 angstroms. And the problem with NMR, one of the problems of electron microscopy is that you have to use frozen hydrated samples to get a sort of undistorted view of the proteins. But these samples create a have a small contrast So the non-protein versus the protein are almost indistinguishable, especially for someone who is not used to look at these kinds of data. But there's a recent development that was able to really improve the contrast, and it has been known for a long time, but nobody could really manufacture a device that would change the phase of the electron beam in a way that would enhance the construct. But that's now possible, seems possible, expensive, but that will probably cause a small revolution in electron microscopy, and we'll see a lot more structure. And then X-ray diffraction, I mean, there are lots of things happening all the time. Methods are being developed further, procedures streamlined. I mean, the possibilities are endless, but I want to just talk very briefly about one aspect, and that is the new machines that follow the synchrotrons as sources of X-rays, and they are free electron lasers. So these free electron lasers essentially are very simple things. They use an electron source where a high energy electron beam comes in between a series of magnets which are causing magnetic fields that are oriented either up or down. So let's say the violet is up and the green is down, and so on. And the electron beam is forced to wiggle around these. And if it's done in the right way, if the geometry is right and the electron energy is right, then this wiggling causes, it will always cause the emission of electromagnetic radiation, but if the circumstances are right, then there is a stimulated emission, that is the lasing action in this caused by this electron beam and the magnetic field. And so the electrons can be easily deflected and leave the scene, and the light beam or the X-ray beam is continuing to the experiment. So this is a very simple thing. It's not as easy as it looks in this picture, and there are machines under construction in, or let's say, precursor machines, precursors to the real thing that is intended to be built. These precursors already exist at the Hamburg synchrotron in Germany, and then also in Stanford, opened spring of last year. And another machine is under construction in Japan, and maybe in other places I just don't know about. But the energy is still not in a range that is the most useful for structural biology. So all they can do right now is doing preliminary experiments, and there was a flood of publications just recently about such preliminary experiments. But the great promise of these machines is that they emit a beam of brilliance, and brilliance is defined here, the number of photons per second per square millirad per 0.1% relative bandwidth. So essentially the strength of the beam. This is the development of X-ray sources over more than 100 years, and starting out with probably Dr. Röntgen's original apparatus. And when I came into this field, we were about here, brilliance of the order of 10 to the 12. And then the synchrotrons brought huge improvements, and note this is a logarithmic scale. So right now we are at this point with about 10 to the 25 or 24 photons per second and so on. And the free electron lasers are promised to deliver at least 10 orders of magnitude more brilliance. So essentially the idea would be, okay, if we now need a crystal to produce a significant diffraction pattern, we just can go back to one molecule and produce a significant diffraction pattern. And that would sort of take out of our lives a major headache, and that is crystallized proteins. And so the idea is, among other publications, is shown in this figure from a picture by Gaffney and Chapner in 2007. It would use the X-ray beam of a free electron laser, and I should say it's very important. A very important point is that this beam comes in pulses, very, very short pulses. And short means in the order of tens of femtoseconds. Ideally, maybe 10 femtoseconds or less. So there's this beam, and perpendicular to it would be a particle stream consisting of protein molecules. And I think similar beams are already used in mass spectrometry. I mean, there are means to create such beams, particle streams. And whenever a pulse and a particle coincide, the particle causes a diffraction pattern. This is a continuous pattern because there's no crystal. But, and each particle would be in a different orientation, so you would get sampling of the orientations. And of course, this tremendous brilliance is causing radiation damage much more than we can imagine now. That means it will evaporate the protein. But calculations show that if the pulse is short enough, the evaporation will set in a significant way only after the pulse is over. And evaporation, one of the reasons is that the electrons a significant part of the electrons are shot out of the sample, and so there's a much excess of positive charge, and that would drive the molecule apart. But that would all happen after the experiment. So we lose the protein, but we can still have the diffraction pattern. And then it would be a process similar to what is nowadays already done in electron microscopy. Just instead of images, picking out characteristic diffraction patterns, averaging them, finding their relative orientation, and essentially reconstruct them. So there are colleagues who believe all this is just smoke, pipe dream, and matters. I mean, a method to persuade the funding agencies to spend money on this, and others give it a really serious chance. And I think I belong more to the latter. So that could be the next development in X-ray crystallography. Of course, it would need a huge number of technological improvements just to think of detectors and but and we are very far from this being a workable method, but that may come. Okay, so the last part I want to talk about is the question: can we make reliable predictions in structural biology? And I will try to explain what I mean by that. And the example is well known to some people here, the transporter FEC-A for ferric dicitrate through bacterial outer membranes. And this is a part of a family which are called TONB-dependent transporters. And I will try to explain what that means. Okay, so quite a while ago, Andrew Ferguson, when he was in my lab, determined structures of two versions of this transporter, FEC-A. One with without a ligand, and another one with a ligand bound to it. And you can see here how they are situated in the membrane, outer membrane of the protein of the bacterium E. coli, and the parts that undergo structural change upon ligand binding are shown here in color. These are two loops between beta strands, and this I should mention, this is a structure that consists of 22 beta strands, and in the center, in the pore that is formed by these beta strands, is sitting a so-called plug domain. And at the outside of the membrane, there is a ligand binding site. Okay, the plug domain has 3 loops which are involved in ligand binding, and there are various helices, and I don't want to go into detail, except to mention that there is, in the unliganded structure, there is a helix at the beginning of the structure. So this is near the N-terminus. It's not exactly the N-terminus. There's another domain here which we cannot see in this particular crystal or this structure analysis. So the chain comes in here and forms a helix. And this helix then gets into the first beta strand and continues then it gets into the plug domain. And on the right, this helix has disappeared, and so there is a movement in this protein, which I try to illustrate here. So if we concentrate on this and that portion, and it's switching between the two structures, you can see that there's a helix disappearing and appearing, and this helix is going away, and loops move and so on. So this is what I think makes this protein. It's one of the things that make this protein quite interesting. And this structure change here has been observed in quite a number of different such transporters that were determined. And among them labs that work on such is Susan Buchanan's lab here at the NIH. And so it must mean something, and I come to that in a second. Here's a top view that is perpendicular to the membrane, and you see here the cartoon of the unliganded and the liganded structure, and the ligand binding site is here. Here's a a surface model with the ligand shown, even though it's not in that structure. And here the ligand is gone because the loops have folded across the ligand binding site. And here you can see these, this TON-B, the switch helix, and here you cannot see it. And that is the here's a cartoon from a review by Volkmar Braun and Marn, and it shows the functional context of this molecule. Here's unloaded FEC-A, loaded FEC-A. The difference is not just that the ligand binding site is occupied, but also that something called the TON-B box, that is a short stretch of 5 amino acids that is known to interact with the inner membrane protein TON-B. So this is the inner membrane, that this is exposed and it's interacting with TON-B. TON-B apparently pulls on this TON-B box and somehow causes the ligand to come through the membrane, either by pulling out the plug domain as a whole or unfolding it by just pulling on one side. And there's another effect, and that is the amino terminus of FEC-A has a separate domain of about 80 residues that interacts with the inner membrane receptor called VEC-R, which sends a signal upon that interaction to activate gene expression relevant to the import of ferric citrate. So not only is the ligand pulled through the membrane, but also more proteins, more copies of these proteins will be synthesized. And the ligand essentially binds to a periplasmic protein and is transported from there through the membrane by an ABC transporter type of molecule. Okay, so that structural change is a very important part of the whole picture, because it is clear that there's no point for TON-B to pull on an empty a molecule with an empty ligand binding site. So that is the signal for TON-B to really interact. And so that raises this question. I mean, ligand binding to FEC-A at the outside causes conformational change at the periplasmic site. There's no structural change in between. So what causes the conformational change at the periplasmic site? And it seems a simple question. It's not an earth-shaking question. It's not that biology cannot go on without answering this question, but I use it as a demonstration of what we still need to learn about proteins, or what protein structures could tell us that we don't know how to ask. And so we used bioinformatics to find a pathway for signal transduction. And this is a method that was developed in the lab of a colleague, Rama Ranganathan. He calls it statistical coupling analysis. And its premises are, okay, there are many different transporters of the type of FEC-A, and they exist in many organisms. Okay, so then if there's a common signaling mechanism, which would make sense, then functionally important residues should co-evolve. And this statistical coupling analysis, using a large sequence alignment in the order of over 500 sequences at the time, should help identify these co-evolving residues. And then mutations of these residues should have measurable effect on the function. And we did that. And here's a short excursion on statistical coupling analysis. So the idea is that energetic coupling of a pair of sites should mutually constrain the amino acid distributions at these sites and should be revealed as the statistical covariance of amino acid distributions. And so if you have a site I, which in the sequence alignment has 50% histidine and 50% valine, and you just take the subset of sequences that have histidine, then you can, if you're observing a high coupling, you would, let's say, or the ideal case would say, okay, these histidines come along only with isoleucine in the site J. So this would mean these sites are perfectly coupled. And vice versa with valine. Or of course it could be that even if you take the subset with histidine, the distribution is still at site J is still the same, and then there was no coupling. Okay, and he tried that on FEC-A, and here is the result. FEC-A molecule, the ligand, so this is the liganded state, switch helix down here, and the blue and red residues are those that are in a sort of a group of statistically coupled residues. And you can see that there is a chain of residues going down through the membrane to two different sites at the periplasmic site. So this would be the periplasmic site. And here are cuts through this arrangement. You can see. And that was pretty satisfactory. We were a little puzzled why there is a bifurcation of this signaling pathway. But then there were, while we were writing this up, there were two structures published which showed complexes of a domain of ton B with two other transporters, FUA and BTUB. And it turns out that in these cases, the ton B domain binds in slightly different places, which are at the ends of the bifurcated pathway, of either end of the bifurcated pathway. And so maybe there's certain residues that decide which way this domain goes, but that's so it shows a little bit that the statistical coupling analysis can give some answer, but of course it does not provide a physical explanation. What are the forces? What are the interactions that make this possible? And so we are tempted now to go to computational methods and perhaps normal mode analysis, but I cannot really imagine that this would give, of course, the normal mode spectrum would be somewhat changed upon ligand binding, or we could do molecular dynamics calculations, and molecular dynamics is a very, very complicated and time-consuming process, and this is because it uses essentially Newton's law, mass times acceleration equals the force, and the force is the derivative of the total energy of an atom. So it's the alpha goes from 1 to the number of atoms, And, and the potential which is used is a function of bond lengths and bond angles and dihedral angles and van der Waals contacts and Coulomb interactions. And so every atom is in the force field of all other atoms, and as soon as this structure changes, which happens in dynamics, the energy has to be recalculated, etc. So that makes it extremely time-consuming. In our case, is would involve in the order of 7,000 to 8,000 atoms. At least, no, it's more than that. These are just non-hydrogen atoms. So it's pretty much outside of today's computer capacities. And I was very amused reading these statements in Physics Today a few years ago. So there is Eugene Wigner, who was a sort of theoretical physicist living around 1960 or publishing this 1960. So the miracle of the appropriateness of the language of mathematics for the formulation of the laws of physics is a wonderful gift which we need to understand nor deserve. Good. That this may be true if you're looking at Maxwell's equations or the Newton's law of gravitation, etc. But then what's more relevant to our case is turbulence, friction, and protein folding, for example, are important, but despite much effort, and I can only emphasize this, they remain largely mathematical wilderness. So we are in a mathematical wilderness with respect to protein structure. And I can only appeal to the young people in the audience. Please try to bring us out of this wilderness. Otherwise, we have to wait for the time when the computers are fast enough to do this tedious molecular dynamics thing. And of course, I mean, some people are jumping ahead, and last year there was this paper which really gave me some hope that something will be happening. This is, of course, making a faster computer, a computer that's over 100 times faster than the fastest next best computer, and specializing it on, I mean, the speed was achieved by hardwiring the calculations of that were necessary for molecular dynamics. And so the David Shaw group, I think for the first time, simulated the folding of a small protein successfully, just by first, I mean, doing nothing but inputting the an extended model of extended polypeptide chain and solvent, and letting it go. And according to their report, they observed the true structure, which had been also solved by X-ray crystallography. But it took about simulation in the order of tens of microseconds or 100 microseconds, whereas the furthest we have ever gone, we, that means my colleague Arne Strand and I, in our sort of innocent attempts to do some calculation, the furthest were about 1,000 times less. And it took us half a year, so 1,000 times half a year is far beyond what I can imagine. Anyway, so either we wait for even better computers, or somebody makes a fundamental discovery how to better do simulations of proteins on protein dynamics. And with that, I close and thank you for your attention.
after.
So Dr. Deisenhofer will be pleased to take questions from the audience. So please go to the microphones.
You made significant achievements. So in terms of challenges, could we improve on the photon reception and energy conversion in food production using the bacteriorhodopsin model you have studied? Is there any way we could engineer it for enhance the food production?
Did you understand the question? No? Okay.
We could not hear the question up here.
So to make more food for 7 billion people in this planet, obviously the source of light has to be converted. So is there any more efficient way? Mother Nature obviously spent millions of years to evolve these photoreaction centers. Is there any way we could improve on it in terms of engineering the proteins?
Yeah, there may be, because what Mother Nature has to look at the complete picture, and that is that, I mean, the organisms have a prime interest in survival, so they have to make sure not only that they do optimal photosynthesis, but many other things. And so it might be possible in a very supportive environment to support a type of photosynthesis that's a little bit more efficient. But so far, I think the efforts go in a different direction, and that is, for example, creating photosynthetic organisms that directly convert the energy into, let's say, synthesis of lipid-like molecules or things like that, which would open the possibility of getting a much better source of biofuels. And unfortunately, it already turned out that this, using, let's say, existing organisms, it's not promising, it's not economically viable. And therefore there had to be development, construction of specific organisms but the changes that are being considered are more on the side of introducing new pathways of synthesis than improving the photosynthesis itself. And so I think there is a lot more leeway for improvements in parts of the biosphere that are other than photosynthesis. Photosynthesis is a very, very complex process, especially in plants where you have this succession of two photosystems, and a lot of things work together, 100 proteins involved in the process. So I don't see an immediate chance for really path-breaking developments there.
Hi, thank you for the talk. I have a question regarding your opinion about observing the real-time in vivo protein structure in cells. What are some technology which would be promising for this purpose? Thank you.
What are the prospects of observing real-time in vivo in cells?
For example, how can we observe the protein movement in the cell instead of in the crystal or in the 2D crystal?
Yeah, that is. I mean, the only technique that I could imagine right now is perhaps NMR, but or spectroscopy. Observing single molecules is always, requires the isolation and purification of these molecules. If you imagine the dense soup of mostly proteins inside cells, then either you get a signal coming from all of them, and then you have no clue what you're seeing, or you get most likely no signal at all. So I don't see in the near future, and that means in the next 10 years or so, a chance for seeing structures in living cells. Of course, there are investigations of cells by electron microscopy, and the big thing now is tomography, where you essentially create a three-dimensional picture of a cell, but the cell is dead by then. I mean, it cannot survive, or it has to be in a frozen environment so that it's fixed, and the radiation damage would kill it anyway. So I don't see much of a, but maybe I'm just wrong. Yeah.
Could you please say just a few more words about what are the specific reasons why some people believe that the free electron laser approach will not work?
Well, one argument I heard, that it will work, but that it will not give us any new insights. And that is because, in principle, you can do the same thing with electron microscopy. If you have frozen hydride sample, randomly oriented molecules, you can record the pictures and average and classify, and maybe end up in the same point. The free electron laser also has a size limitation, because in order to see an informative diffraction pattern, you have to have a large enough molecule. And I don't know exactly where the size limit is, but it's not that you can just send a sort of a carbohydrate or a sugar molecule in there. You have to have probably 100 kilodalton or so. And so the similarity of the process in electron microscopy and in potential application of the free electron laser, I think causes most of these cautious voices. And then, of course, I think that there is a chance that by removing the background from the scattering through water, that one can see much more clear structures. And then on the other side is the argument, okay, electrons interact with matter a lot more strongly, many orders of magnitude more strongly than X-rays do. And so why use X-rays at all? But that is a little bit against my, how do I say, all I did during my career. So I think there is a good reason to use X-rays. So I don't know whether that answers your question, but it's a situation that resembles the time before any major experiment. And then it's not clear what will come out. Maybe there are unexpected difficulties. And so they are the half full people, half full, glass half full people, and the glass half empty people. So I think.
Okay, well, let's thank Dr. Deisenhofer for an illuminating lecture. And I'd like to invite you all to a reception in the library, and there'll be an opportunity to ask more questions that you may be too shy to ask in public. It's very nice.